Okay, so it's 4:32. So I'm just gonna get started, and so people can trickle in, and that's fine. So first of all, I want to welcome everyone again to this Bay Area Microfluidics Network event. We're really excited to have you. We're really excited about our speakers today. Like Samira was saying earlier, for those of people that just joined now, this is an event that we've actually wanted to do for a long time. So for 2 years, we've been wanting to do a microfluidics and space event. So we're very, very excited for today's event. And of course, like, thank you all for joining and making this event possible. A little bit about the Bay Area Microfluidics Network for those who are not familiar with us. So Bay Area Microfluidics Network is run by 5 people, and it's just on a volunteer basis. And those people are Thomas, Adam, Samira, Anushka, and myself. And you'll see them in today's event. Some of them might be trickling in a little bit later. But in just a sentence or two, Bay Area Microfluidics Network, what we aim to do is to connect people. That is, to connect people in academia and people in industry. And we want to kind of like foster this like network of microfluidics, especially because microfluidics is so present in the Bay Area, right? And dealing with the topic of Bay Area Microfluidics Network, I wanted to talk about just 2 initiatives that we're starting to get the ball rolling on, and we would love for those people who come to our events to also participate in these initiatives if they can. And the first is the BAM, the Bay Area Microfluidics Network mentorship program. So we talked about this at our last event, but this is just it's a mentorship program that we would like to start, where we can connect those people who have been in industry for a long period of time with people who are kind of like just starting in industry, or people who are still in their PhD programs, but would kind of like want to learn more about what it's like to have a career, right, in microfluidics. And this program is informal. We would like mentors and mentees to meet twice a year, and of course they can meet more often if they desire. But we want to start this in around September of this year, right? So it's a couple of months away, and we're currently looking for mentors and mentees. And if you'd like to sign up to be a mentor, if you'd like to sign up to be a mentee, here's the link. I think Thomas is going to send it out to the group chat, so you can click on the link and you can sign up. Also, if you think if you think you know someone who would be a good mentor, you can also write that person's name in the form, and the BAM leadership can reach out to them. So please let us know if you're interested in this program. We're very excited about this. This is another initiative that we are already doing, and a lot of the people that are on this event have probably joined these events before, the Friday Breakfast Club. So these are also informal chats that we do between an industry leader and around 9 participants, usually PhD students, sometimes other people in industries. Sometimes we have more participants, but the idea is just to have coffee with someone who knows a lot about microfluidics on a Friday morning. And we do these every so often, sometimes once a month, sometimes twice a month. And we're looking for leaders to invite as featured guests. So if you would like to be the leader invited for our Friday Breakfast Club, and all of these events are on Zoom, by the way. If you would like to be a leader invited to a Friday Breakfast Club, or if you think you know someone who would be a really good leader, again, please express your interest. And here is another link where you can submit someone's name. And I think Thomas will also be sending them out. Finally, if you haven't already, you should check out our website, bayareamicrofluidicsnetwork.org. And if you have any questions about any of the initiatives, any of the future events, or anything else regarding Bay Area Microfluidics Network, don't hesitate to send us an email. And you can private message us also during the Zoom event. Thomas, Samira, Anushka, other people will be trickling in, any of the leadership, and you can ask, and we can put you in contact. I think a lot of you have also seen our LinkedIn. If you haven't, just search for us on LinkedIn, and it's a good platform to share job openings, interesting articles, etc. in microfluidics. And this brings us to tonight's program, where we'll start with this introduction as soon as I'll be done in a little bit. And we'll start with Tony Rico, who's joining us from NASA Ames. He'll be the first speaker. He'll have 20 minutes, followed by Dr. Nathan Brommel from Lead in Measurement Technology. And then finally, we'll have a moderated discussion moderated by our own Don Arnold, who's done a great job of moderating the discussions before, and I think he'll do a great job again. Finally, we'll do networking in breakout rooms, and I think between these two events, we're going to make some announcements about open job postings that have been shared with us. This is just a little Zoom etiquette. Most of you should remain on mute until the networking session. Okay, that's until 6 p.m. But if you have questions, type them into the chat and send it to everyone. And Dawn, during the moderated discussion, right, Dawn will be looking into the chat and seeing if anyone has any questions. So you can just send them to everyone, right, on the Zoom chat function. And also, if it would be helpful if you change your name to your first name and your last name and your affiliation, just so we know who's represented at tonight's event. Finally, for the networking session, this will be in breakout rooms. You feel free to switch between the breakout. There will be a breakout room for each speaker. So that's Tony, Nathan, and Don. And feel free to switch between the breakout rooms. And if you don't know how to do that, you'll be assigned to a breakout room, and then just go to the main room and just message into the chat, and one of the leaderships will help you out. So I hope that makes sense. Besides that, the only requirement is to stay engaged and have fun. So without any further ado, I'd like to introduce our next speaker. So Tony Rico is our first speaker for tonight. And if you give me just 1 second, I'll read his introduction. Great. So Dr. Tony Rico received his BS and PhD degrees in chemistry from UC Berkeley and MIT, respectively. He's held positions at Sandia National Labs, the University of Heidelberg, Aclara Biosciences, the Biomedical Diagnostics Institute in Dublin, Stanford University, and NASA Ames Research Center. His R&D experience includes chemical microsensors and microsystems, polymer microfluidic systems for biotech research and pathogen detection, point-of-care medical diagnostic devices, integrated autonomous bioanalytical systems for space biology and astrobiology studies aboard small satellites, and search for life analytical payloads for missions to the icy worlds of the solar system. And with that very interesting introduction, I'd like to give the floor to Tony. And Tony, you can go ahead and share your screen if you want to show your presentation.
I sure will. Thank you. Let's see if this works as a presentation. All right, how does that look? Is everyone seeing that?
We can see it.
All right, good. So thanks for the kind introduction. Really, really nice to be here with you today. I've been, as you heard from the introduction, I have roots that go way back in the Bay Area. Besides getting my undergrad degree at Berkeley, I actually was born in Oakland, so there are a few of us locals still left around the Bay Area. And my start in really microfluidics technology was at Aclara Biosciences, which was located in Hayward when I started, and then we moved to Mountain View. And I was there until I moved to Stanford and NASA, where I continue to do various things in microfluidics. That's what I'll tell you about today. The talk's called Life in Space. And so I'm going to tell you first a bit about how we use systems that have microfluidics at their core to study terrestrial microbes that we carry into space and see what happens to them. And then how we've adapted some of those microfluidic technologies to go and look elsewhere in our solar system for the presence of life, particularly biomolecular indicators, but particles and so on as well. And I think Nathan may tell you more on that side of things. Over at the left here is a little fluidic block that's in a way large, probably used to what, compared to what some of you are used to working with in microfluidics. It's about 4 or 5 centimeters on a side. But this is not only some microfluidic networks and a bunch of wells for growing bacteria, but it also includes all the optics, the heaters, the electronics, the mechanical structure, some radiation sensors and everything. So it's maybe more integrated than what some folks are working on, though I know integration is much more common than it was when I started in microfluidics. And it's a little bigger. For a spacecraft instrument, though, it's still very much on the small side. Over at the right-hand side here is the kind of fluidics that we're using to develop our search for life technologies, and I'll talk about those more later. So quickly before we dive into the technology, I just want to point out that there are these sort of two different areas of biology and space, and I'll talk about both today. Astrobiology is very fundamental. It's looking at what are the origins, evolution, distribution, and the future of life in the universe. And that includes things like the chemical building blocks of life. Can life survive elsewhere than on Earth? And where else might we find life? Fundamental space biology is actually what I'll tell you about first a bit, and the way we study it with microfluidics and very small satellites. And so that involves taking terrestrial biology into outer space and looking at the two primary perturbations when we go into space, less gravity than we have here on Earth, and radiation that's typically ionizing and significantly higher dose than we're used to on Earth. And biochemical effects of the extraterrestrial environments are of interest as well. And sometimes all these effects synergize, and sometimes they're antagonistic. And why do we do that? Well, in part because that helps us understand what humans have to deal with when they go into space for missions, or someday maybe for longer term habitation, but also because we can learn things relevant to, for example, healthcare and aging on Earth. If you go microgravity as an astronaut, you begin to lose bone density in the heavy weight-bearing bones of your body, your pelvis, upper leg bones, at an astounding rate of 2% of bone density a month, a little less than that. And so that's really accelerated aging. You know, if you're an engineer, you know accelerated aging is an important thing to be able to do, understand how a system degrades over time, and to do it without taking the full lifespan of the system. Well, losing bone density at 2% a month, that's pretty drastic compared to what happens to all of us as we age and typically gradually start to lose some bone density. So by studying that, insights have been able to be gained on the process of osteoporosis and how bone gets rebuilt. This is a slide that summarizes a number of what we call nanosatellites. And if you're not familiar with that term, it simply means a satellite that's smaller than 10 kilograms in total mass. Actually, sometimes that ended at 14 kilograms for reasons we don't have to talk about. And that came about because for a while, the satellite industry called any satellite smaller than 100 kilograms a microsatellite. So very different than many of us know terms nano and micro in engineering. But anyway, you see this satellite over here. It's about a foot, 30 centimeters long, and about 10 by 10 centimeters in the other two dimensions. So it's certainly not micro in size, but it's pretty small as satellites go, and it would weigh something like 5 kilograms. So these are the different sorts of microbes that we have taken into space or plan to take into space to study in some of the missions that we have flown. E. coli, we launched our first mission with biology in what's called a CubeSat here. This is a so-called 3U CubeSat, 1U being a 10 centimeter cube. So you stick 3 such cubes together and put solar panels on the outside, and you call that a 3U CubeSat. We launched that CubeSat in 2006, and it let us study gene expression in E. coli in microgravity in Earth orbit. More recently, about 4 years ago, we launched E. coli antimicrobial sat, ECAMSat, same E. coli. We were looking at antibiotic resistance in the microbes that affected their longevity, and why was that different in microgravity? So the microfluidic card that was the basis of this second mission, and it has 48 wells, 100 microliters. Again, a big quantity compared to what you might be used to in microbes, but cultures of microbes sometimes need to be a larger numbers so that they can have appropriate quorum behavior and so on. We launched something called PharmaSat in 2009. This was to study drug dose response in Saccharomyces cerevisiae, one of the world's most important organisms, maybe the most important microbe on Earth, because it lets us make beer and wine and bread. So where would we be without those? And looked at how drug dose response is affected by microgravity, and there is some effect there. We plan to launch towards the end of this year into interplanetary space. I'll say a little bit more about that in a moment. Oreos was a satellite I'll also mention a little more momentarily. The organism and organic response to orbital stresses. That launched in 2010, and that looked at survival and metabolism of microbes, and also did an optical experiment looking at thin films of organics that could be biomarkers or organic building blocks. We launched SporeSat in 2014. It had a series of ion channel sensors to look at this fern spore, aquatic fern spore, Ceratopterus ricardii. They have the property that they actually have gravitation sensors built into them. And when they detect gravity as they begin to germinate, they send a flow of calcium ions out the top, and calcium influx happens through the bottom. And those two phenomena can be measured across this 100 micron spore if you have a pair of electrodes on either side. So that's what we did in these little tiny centrifuges, 50 millimeter diameter centrifuges, where we could vary the gravity by the rotation rate and look at differential calcium ion concentrations. Finally, a microscope that's sort of a predecessor of some that we're working on and Nathan is working on these days for doing dual wavelength fluorescence imaging. And this was to fit also in one of these same footprint small satellites and to look at the roundworm C. elegans and look at how different tissues, different cell types in them are affected by microgravity. So taking you just briefly through how one of these missions is put together, GeneSat. This was, as I said, the first one. You start with the organism, figure out the microfluidic home. In this case, it had to have fluidic access to the wells and good optical properties so we could make sensitive fluorescence measurements. And that gets combined with a fluidic system here. There are some small electrically actuated solenoids, little bags for pumps and so on. And this whole thing is about, 15 centimeters long, so not very big. Into a hermetic container. It's got, of course, all the electronics, optics, everything built in there. So this is truly integrated. This is not a chip in a lab. This is a lab in a can. And then you've got the rest of the satellite at this end, the power, the communications, the command, the data handling, telemetry, and so on, little antennas sticking up here that you can't quite see. And this goes into space after 6 weeks of being on the launch pad as a secondary payload on this rocket and winds up feeding the E. coli, growing them. And now you can see when the data come back, how do the organisms grow differently in space flight than they grow on the ground? And the short answer is they grow a bit faster on the ground in this case because there's more gravity-driven convection. So let's see, where are we going next? Well, we're going into interplanetary space. This will be the first biology experiment ever to leave the Earth-Moon system when it flies, BioSentinel. And it's also a cube satellite. It's a 6U cube satellite, so twice as big as a 3U. And it will take us from what we know, which is sort of out to the distance of the moon and durations of as much as 15 months in the space station, and go out to distances comparable to how far away Mars is. BioSentinel will end up in an interplanetary orbit, an orbit around the sun, heliocentric orbit, and never encounter Earth again. So it'll be in orbit essentially forever. What we'll do is use yeast on this satellite to look at DNA damage and repair due to the radiation environment, principally in deep space. And this is sort of an exploded view of all the various different components and parts of it. The part of most interest to me and where the microfluidics are is in here, the biosensor payload, which has 288 microfluidic wells with little cultures of yeast, and they start at different time points to grow when they're out there in space, and we measure them optically. And then you see all the other parts that are clustered around this in this small package. And the whole thing weighs, when it's all put together, about 14 kilograms. And so that'll go to deep space along with the Artemis 1 spacecraft. So let me jump in the last couple of minutes into how we're searching for life. This is some of the multi-layer fluidics that makes up the heart of the BioSentinel fluidic system. And these are three precision machined layers of polycarbonate that are thermally fused together and make up the wells and hold the valves and then interface with the electronics and integrate everything. Little cute little things here like a desiccant trap, check valve, and so on. And you can see the scale of this. And so this is sort of a multi-layer printed circuit board for fluidics, again, familiar to many of you. So when we search for life, we do something called following the water. And there is some water on Mars in the form of frost, but if you want to really look where there are oceans, go to Enceladus or Europa, which have oceans. In fact, Europa's ocean has more water in it than all of Earth's oceans combined. But it sits underneath a thick crust of ice. Enceladus also has a quite substantial ocean comparable to a fraction of the Atlantic Ocean volume. So those are places where we might look for life, where there are salty oceans to be found. What do we look for? We look for a combination of factors that hopefully are agnostic in terms of telling us if life is present or not. Chemical building blocks you need to make life. You need some way of storing information. So that would be multimers, polymers, some structures for containment of life, molecules that do particular functions like transferring energy or transferring chemical species, transferring electrons. And if you found all of these by analytically looking for each of them, you would be pretty certain that this wasn't what you would see if there were no life present. And so it's really about comparing what's the signature we find with what would we expect to find if there were not life present in a place like Enceladus or Europa. So to do that, the job of the microfluidics is just to do a few things. And again, many of you are familiar with lots of these. Basically, grab the sample and then do things like pulling out particles, which we might look at with a microscope, degassing, changing the ionic strength, adjusting the pH, adjusting polarity, concentrating, maybe diluting, making standards and reagents. And which of those we do all depends on what instruments we're using to do the analysis. And you see some of the ones that we're looking at, some of which we're developing ourselves and others of which are developed by our partners. And so we don't necessarily do all these fluidic operations for every instrument. We do the ones that each one needs, but that's familiar to a lot of you. And so the idea is the sample comes in here, we could look at the particles with a microscope and then do a sequence of processes in the microfluidics to make it ready for capillary electrophoresis, mass spec, some electrochemical sensors, ion selective electrode array. This manifold, which does most of the processes you see here, is one of the first ones we developed a few, was aimed at addition to Enceladus. Here's a microscope, and I don't know if Nathan will tell you more about this one or not. He's building the microscope part of this. We're putting together the fluidics part of it. And basically this takes a sample that would be delivered to us by a Europa lander, melts it, processes it fluidically, and spills it out onto some filter membranes that are silicon nitride with nanometer scale holes in them where we capture the particles. And this disc is fluidically connected so that we can direct the particles to the appropriate filter, but it also rotates in order to put the appropriate little silicon nitride filter membrane here underneath the microscope so we can look at each one of the filters in turn. And this ultimately will look at 7 different samples, and for each of them look at 3 ranges of particle size. Everyone's familiar with bubbles. This is our bubble trap generator, and so this is more sort of a customary device for many of you. The bubbles go in and they just disappear. But we also use the very same device by plumbing it differently as a bubble generator, and so we can pull a vacuum, the hydrophobic membrane that generally acts as the bubble eliminator and create bubbles instead. And the reason we want to do that is this bubble also is a decent electrical insulator. And this is part of a system designed to do capillary electrophoresis, which is a good way to differentiate left and right-handed molecules such as amino acids. And so the reason we do that is this fluidic system is connected to 8,000 volts to do capillary electrophoresis. And the bubble is a nice way to keep the electricity out of the salt water and therefore not blow up our fluidic system. The last instrument I'll tell you about is the microfluidic icy world chemicalizer. And this is an array of ion selective electrodes and other electrochemical sensors based to a fluidic manifold that has various pumps, valves, and so on. If people want to ask about those, I can walk you through some of them. And again, it's fairly small and integrates a lot of functionalities and serves 48 different electrodes. So that entire system fits in a box that's about 20 by 10 by 10 centimeters. Again, familiar size. It could be a CubeSat, but in this case, that fits in one of the slots that's been designated as a possibility for a Europa land to go for signs of life on Europa. And with I'll say thanks to all of you and to the several villages worth of people that have helped these projects. While you see lots of names here, they represent a wide diversity of backgrounds and interests and expertise. But I'll say that on most of these projects, usually at most one or two of the names on here, people are full-time on them, and all the rest of us are working 2, 3, or 5, or like Nathan and me, 8 projects in parallel. And so you need all of us both to get enough time, but also to get the diversity of experience and so on. I guess my bottom line here is that microfluidics at NASA for these kinds of instruments is really, really diverse in all the different fields you have to bring together. And it's not just a few of us sort of microfluidic engineers, but you need everyone from the biologists to the software people and everyone in between. So it's all about the integration. Thanks a lot. Happy to take some questions.
Samira, are you ready to go?
No, I was just going to say that people can ask their questions in the chat, or feel free to unmute yourself, or and ask your question.
I think the plan is to have questions during the discussion section. So, so by all means, ask your questions in the chat. And then after Nathan's talk, we will ask all the questions of the speakers.
All right.
So
I see names I know. Nicole, Ben, hey guys.
so thank you very much for that. Thank you very much for that, Tony. Samira, would you like to introduce Nathan?
Yeah, sure. Thanks, Tony. Yes, pleased to introduce you guys to Nathan, the founder and CEO of Light and Measurement Technology that develops novel scientific instruments and microfluidic platforms designed for extreme environments on and off the Earth, such as the South Pole, Greenland, and Europa. For the past 15 years, Nathan has focused on creating instruments enabling the search for life and characterization of organics in our solar system, working closely with NASA, with people like Tony and other university and private sector partners. So without further ado, Nathan, please take it away.
Thank you. Can everybody see my screen?
Yep.
Great. I just wanted to thank you for this opportunity. And I know I was a last minute replacement. Joran Haring was supposed to give this talk, but he was not able to just a short time ago. So without further ado, I'll get started. I just want to give a brief outline. I'm going to talk about how I came to work in microfluidics. My background is a bit diverse, not typical, and coming into this field. And then I'll talk about what we at Light and Measurement Technology do in microfluidics, and sort of perspective of opportunities for businesses beyond space exploration who want to participate in this space. So I began my studies as a physicist, and I worked at UC Berkeley. And as part of my graduate dissertation, we were actually doing paleoclimatology using borehole optical logging and developing strategies and instruments to detect life immured in glacial ice. These were actually spin-off projects of neutrino detectors down at the South Pole. But they sort of formed the foundation for developing things that were rugged and reliable and in small spaces. I actually went to work at NASA Ames as a postdoc in the postdoctoral program. If anybody's interested in working in space, I highly recommend that program. While I was there, I got the opportunity to work on a lot of great projects, including the Orioles nanosatellite. That's where I met Tony. I also worked in astrochemistry, worked on snowpack ecosystems, studying snow algae, and biomarker instrumentation work. After that, I guess my first real job was actually at a small company called Los Gatos Research, which is primarily known for cavity and hat spectrometers for gas phase detection. They've recently been purchased by ABB, but if you're interested in that, I would highly recommend them as a vendor. But this is where I actually got my first taste of microfluidics. I was given an orphan project to develop a microfluidic system that uses capillary microchip electrophoresis as detection. And the whole goal was sort of to take the lab in a chip and put it into a lab in the box. And this was the first instrument. It's known as the chemical laptop that I worked on, and it was delivered to JPL. If you're not familiar with the capillary electrophoresis and the microchip version, you basically work with a cross-channel chip. You inject your samples through the two little side arms. That creates a plug. You apply a high voltage. That causes the plug to rift and the different constituents to separate, at which point you detect them towards the end of their travels. The detections change. This one was lift detection, and it was designed for amino acids. And it's been working down at JPL for the last, well, better part of a decade now. And the technique is actually quite sensitive. You can get under 200 picomolar and better sensitivity of amino acids. And this has been a great opportunity for me to actually get into this field. Shortly after that, I actually went and founded Light and Measurement Technology back in 2014. And we are a small business in Sunnyvale. We focus on ruggedized, miniaturized, automated instruments that go from sample to data. That's sort of our philosophy. We work closely with NASA and other public and private institutions to develop chemical sensing and life detection instrumentation, and with a really good goal on space exploration. Our team is really multidisciplinary, and our research staff consists of people with diverse training with mechanical engineering backgrounds, physical chemistry, physics, and biomedical engineering backgrounds. We do a lot of different things using the tools of microfluidics, spectroscopy, and optics. So why microfluidics for planetary exploration? Well, Tony just talked about a lot of great applications for it. In general, it's small sample requirements. You may not necessarily have a lot of sample to work with. So building instruments with microfluidics is a great way to solve that problem. Smaller reagent requirements. The other problem of sending an instrument that does chemistry off-world is that the reagents can be quite cumbersome and massive. If you can shrink those requirements down, that is great. Microfluidics solves that. Compact hardware, check. Can incorporate complex chemistry in small packages, that can be done. And it also has low power requirements. These are all requirements for space exploration that microfluidics meets. So some applications of microfluidics in space, everything Tony mentioned. Broadly speaking, chemical analysis, you can characterize the environment, which is really important for understanding what you're measuring and what you're seeing. Organics quantification, if you're looking for life, that's an important thing, but also just understanding fundamental questions such as the formation of the universe is very important for understanding organics and their distribution. The life detection is one of my favorites, and microfluidics is really well poised to do that as well. Another thing is sample extraction and preparation. Tony's been working a lot on that as well, and he mentioned that. But you get to work with small samples in microfluidics. And you can prepare these samples for use in a variety of instruments. Because you're working with small samples, small reagents, small quantities, you can fit a lot of different processes into one small box. Tony's been working on that a lot in SPLICE. The other thing is human crew health monitoring. As we get off world and we're talking about building Mars and lunar bases that will be manned and humanized, I guess, people will have to have some way of monitoring their health. Microfluidics would be a great way to do that because they don't need a lot of reagents. And so you can have a long-term duration health monitoring program without a lot of impact to the mission. So some of the things we've been working on at our shop is microfluidic microchip capillary electrophoresis instrumentations. We've been expanding off of the chemical laptop. This is an instrument we have developed and sent to JPL. It's similar to the chemical laptop, except instead of using laser-induced fluorescence, it uses C4D detection. And C4D detection is a means of actually measuring the AC impedance of a fluid as it goes past two electrodes that are printed on the surface of the microchip. In the detection limits of the system that we built are actually quite good. It can detect sub-micromolar levels of sodium, magnesium, and calcium and other ions. We are actually limited by the contamination, residual contamination of the JPL's DI water source and our measurements. Oops. Another MCE system we're working with is as laser-induced fluorescence detection. We're improving on the chemical laptop in that we're actually building chips that have integrated optics on them. There's a number of issues with using standoff optics in the system. They become a little less rugged if you have to align something to the microchannel chip. And if you have to replace it and put it back into the system, realignment always becomes an issue. So using this system improves things from the rugged point of view. We're also expanding the system to use up to 4 different laser wavelengths and emission bands, so you can accommodate a lot of different chemical tags and processes. So you can target a lot of different things if you want to. That will allow for amino acid quantification, chirality analysis, the amino acids. You could use it for DNA with double and single stranded. You could look for a lot of different small molecules. Basically, if you can tag it, you'll be able to use it here. Another system we're currently working on is a sample extraction and preparation system. Our preparation system uses solid phase extraction methods to isolate organics from dissolved inorganic ions, such as salts in the form of brine, other things that could interfere with instrumentation down the stream of analysis. But it also can separate organics into different classes. You can get your non-polar organics in one column, polar organics in another column, so you can send those off to the different instruments that would be measuring them. By using the solid phase extraction, you can actually concentrate your organics by quite a bit, up to 50 to 200 times. Is what we're getting. So you can get a good concentration out of here, which is very important because often the organics loads that you're looking at in space exploration are quite low. So they're beyond detection limits of many instruments. So concentration of these organics is really important. The system can also extract whole cells before they go into these columns. So you can actually, if you're looking for life and you hopefully find cells, you can find them separately. In addition to that, we're developing a front end that also uses ultrasonics to extract organics from complex material off of mineral surfaces, because separating the organics from a liquid brine or something is one phase, but you got to get them off the minerals before you do that. Under development, types of things that we're working on, you're developing methods for using bead assays to quantify different chemical species. So suspension arrays used in space. We're also looking to integrate electrical impedance spectroscopy into our systems, so we can get a lot more information from those. For concentrating very small volumes of organics, we're also exploring the use of ion concentration polarization. You can get a continuous flow of very small volumes and concentrate the organics that way. And we're also looking to develop autonomous ocean and freshwater systems for the long duration monitoring of total organic carbon, nitrate, and phosphate. So a lot of these systems here, the ocean and water systems, they actually require a lot of chemistry being performed on board to do all the reagents, all of the measurements, the grease reaction and all these other things. And so microfluidics is really great for this because you reduce your onboard reagent requirements, which means you can leave it out in the ocean for longer. So one of the questions about space companies and being a company working in the space is scalability and growth. So opportunities to deploy instruments in space can be limited. There aren't missions going up all the time, and we're hoping to get on at least one mission soon or then later. But in the meantime, there are a lot of markets on Earth to target. So a lot of the technologies that are being developed for space, that we're developing for space, have a lot of the same requirements on Earth. It includes environmental monitoring, process monitoring, point of care applications, because they all require small size, automation, low reagent usage, and small sample volumes. So working in space, one of the ways you can keep your company running is by bringing it back to Earth. So these market opportunities could include water monitoring. There's environmental water monitoring. There's also going to be an increasing need for drinking water in water treatment plants to monitor for organics, especially if we have to go closed cycle on board ships, or even in places that are running low on water and want to start recycling their water and back into the drinking supply. Environmental nutrient analysis is very important, not just for scientific purposes, but also in studying algal blooms and other environmental impacting things that can affect humans directly. Point of care medical devices, we've seen an explosion of those in the last year. They'll be a lot more important as well. There are a lot of other biomedical applications, of course. But one other thing that has kind of come up as a possibility is, as we develop these instruments for space, OEM boards and modules that we've developed for applications in space can be very helpful for research on Earth. So that's another market that a company like ours can explore. So speaking of these spin-offs, we are currently spinning off a new company focused on point-of-care medical diagnostics. We just graduated from the University of Toronto's Creative Destruction Lab class of 2021 in their space stream. And this core technology that's going to be funding this was actually developed at LMT, and it will enable large multiplex assays to test for and identify STIs and their drug-resistant strains. So I wanted to close by thanking you and point out that we're always looking to work with new partners and explore new opportunities. If you're interested, please get in touch.
Yeah, thank you very much for that, Nathan. That was really great. So just to, so everyone knows, we're going to take questions during the discussion section, which is coming up shortly. I think Don's going to get us started with let's get the conversation started with some prepared questions, and then we'll open it up to the audience. Thank you to everyone who has already posted questions in the chat. Please keep those coming. So with that, I will introduce Don. So Don Arnold received chemistry degrees from Cornell and UC Berkeley. He began his microfluidic career in 1997 as a senior member of the technical staff at Sandia National Labs. In May 2000, Don co-founded Exigent Technologies to develop and commercialize electrokinetic and microfluidic systems for the life sciences. Exigent was acquired by SciEx Danaher in 2010. During his 17 years with Exigent and SciEx, Don held several leadership roles that included responsibility for technology and product development, intellectual property management, open innovation, business development, and general management. In 2017, Don founded Veristad, where he provides technical and business consulting for life science companies with an emphasis on microfluidics. Since mid-2020, Don has also been an executive in residence with General Inception, an institutional co-founder for science-based companies. So with that, we'll let you take it away, Don.
Okay, cool. Let's see, I've got mute off, and thank you, Thomas, and thanks to everybody for coming tonight. This is pretty exciting space. We've heard two great talks, and especially thanks to Tony and Nathan for kind of giving us lots of stuff to think about out there. And I was particularly excited to see the Saccharomyces cerevisiae going on, so that way when we get to Mars, there'll be a microbrewery waiting for us that's very efficient. Excellent. So cool. Okay, so I think probably a good, interesting place to start is the kind of concept. Tony mentioned it, that a lot of times microfluidics early days, and I think we were around about the same time. He was over at Aclara. We're doing chip in a lab type stuff, and the challenges getting to lab on a chip. And I think one of the great things about this theme that the organizers put together for microfluidics in space is that you've got to design a system where there's really no power cords to plug into the wall. You've got there's no manual sample prep you can do. You can't really go back and debubble by hand. We saw Tony fix that one. What's the what do you do in terms of do you have to end up building a bunch of redundancy, and when these things actually get launched, how long does it take to get a system so it's it's at the point where it's going to be successful without failing? And you know, what's the success rate so far that you've got for microfluidic devices going out in space?
Okay, I guess, shall I start?
Yeah.
So success rate, we're kind of, I think we're 4 out of 5-ish. And the fifth one actually was not a complete failure. In fact, all the ones that have had microfluidics have been a success: Pharmasat, GeneSet, Oreos. And there was another one, a small payload called Power Cell. It was part of a German satellite, and then ECAMSat. So actually there really are 5. The sixth one that had a partial failure was the one I described to you with the little tiny SporeSat, and its failure was that it ended up having a longer stasis period before it could get launched than it was really nominally prepared for, and so the little furnaces were not viable anymore when they got to space. But at least the little centrifuges, the ion selective electrodes, and the control and all the data, everything else worked on that one. So in a way, it was a success, but not a complete success because the biology didn't live. So how do you take? For the ones that have been done more quickly, we basically started with the concept, and about 2 years later, and Oreo is the one that Nathan and I worked on together, might be the best example. We started at in August of 2008, and literally kind of conceptualized and began to design its 2 payloads in August of 2008, launched the entire satellite in November of 2010. So, and that's quick. We've been told many times by others at NASA that you can't do this, it will fail. When they get up there in space and they work, then they say, yeah, this is great that we've done that. So yeah, everyone's a skeptic until it works. One of the things about doing the CubeSat format is that they don't cost as much as bigger missions, and so those smaller CubeSats, they're still multiple millions of dollars. The Oreos one was probably, all costs considered, $4.5 million or something. But that's inexpensive to go to space and do a couple of fairly sophisticated experiments that went on for 18 months, actually, in the case of the optical experiment that Nathan and I were both part of. That's not terribly expensive, but at those prices, you can actually afford for some of them to fail. When we launch our satellites, we think there's about the number varies, but we do risk analysis and say there's an 88 to 93%. We're sort of in that range typically, around 90%, maybe a bit better chance that they'll be successful. And that's, of course, totally unacceptable for anything that carries humans, for anything that's a billion-dollar class mission to another planet. But if you're spending a few million on small satellites and you're doing a number of them and you sort of amortize that over a number of missions as we have, it ends up being pretty acceptable to have sort of a 90%, 93% success rate. So no, you can't build in a lot of redundancy. There just isn't room for it when you're trying to pack this much functionality. We always put 10 pounds worth of function in a 5-pound can. And so that has various interesting names, but it means what you try to do is analyze where's the risk of failure the highest. And if you can, you make that thing or those things redundant. Everywhere else, if you can get the risk of failure down to a fraction of a percent, then you can't afford redundancy. So that's it. There's a significant amount of risk analysis and assessment of failure, and just the things that are unlikely to fail, just don't worry about them failing. Just make sure you do them right. So yeah, it's challenging, and bubbles are always a concern. But you would learn to look a lot at interfaces, and interfaces mean where electricity meets fluidics or where optics meet plastic or any of that. But interfaces also mean anytime you have a water-air interface, a water gas interface, that's something you look at carefully. And in particular, the rule of bubbles in microgravity is it's sort of a corollary to the peanut butter always falls with the jelly side down. My rule of bubbles in microgravity is they go to the location where they completely wreck your experiment. So you can't afford to have the bubbles. You have to manage them either by eliminating them or making sure that they're going to be formed in a place where they can't get in the way. So that's
Kind of
important.
consistent with terrestrial bubbles too, though, right? Yeah.
It is. They tend to do that, but you can
In the
more
corners
predictably
and stay
say
there.
they'll go up in the terrestrial system. Not
Yeah.
always. They go downward in Guinness, but in most cases, bubbles go up on Earth. But in microgravity, they can go in any direction. Of course, they're dominated by surface forces and surface tension and movement of fluid. But that's really hard to predict accurately at sort of these intermediate scale fluidic device volumes.
So did the yeast get you upward moving bubbles in the Guinness then? Is that the goal?
No, in Guinness, the fact that there's nitrogen and CO2 results in a phenomenon where the nitrogen bubbles, which are much smaller and therefore less buoyant, actually get dragged down the outside of the Guinness glass. So if you watch a glass of Guinness settling, you see what looks like stuff falling down the outside, and it's actually bubbles of nitrogen being dragged down the outside by fluid that rushes up in the center due to the CO2 that's carrying it to the top. Really cool fluidic phenomenon.
I'm gonna have to try this after the session.
Yeah, get a Guinness. Try it.
Cool. So let me switch over. Hey, for Nathan, I had kind of a follow-on type of question. You had in your talk, you had some really nice sets of things addressing problems like sample prep and getting samples dealt with and such. I think about the missions that are going and you're out there. Perhaps you're in a place you don't have gravity, you don't have a vacuum, the temperature may be completely out of whack. What about the concept of getting the sample, from the environment? So like if you're going, maybe not just out in outer space and you're not sending up the bio critters or the samples up with it to see how they behave in space, but you're trying to pull in a sample and get it in. Seems like a pretty complex problem there. Is there a lot of work trying to figure out how to pull a sample and then get it, especially if you're looking at trying to get things into a liquid sample? I thought about it with the liquid air interface in an extraterrestrial place where they may have different gases or no gas whatsoever. How much goes into the development of those? And I can imagine them being very different depending upon what the target landing site is.
So they have had some sample return missions where they landed on asteroids and they've grabbed soil. In fact, the Japanese, I believe, sent the first one, and I'm just blanking on names right now. Maybe Tony can fill them in. Yeah. But NASA just landed on just a couple of months ago. They landed and took off, and they're on their way back with the sample.
Okay. Yeah, OSIRIS-REx was that mission. Yeah, it used the vacuum cleaner to pick up the sample. Yeah. Yeah,
and before then, they had Stardust, which was an orbital system, and then they were chasing comet dust out in the solar system, and then came back. They captured comet dust particles in aerogels. These are very complicated systems. One of the things that makes them complicated is if you land on a large body, you need a lot of rocket fuel to get back off that body. So they've been targeting orbital flybys to comet tails or landing on small asteroids. If you go to Europa or Mars, it's a very difficult process to get something returned. NASA's been talking about sample return from Mars, but it's a very long process. They have to build systems that can generate fuel there or get lots of fuel so they can get a rocket to take off back off the surface.
Yeah, and I was thinking about the other situation perhaps where you've sent up an analytical instrument, and rather than bringing the sample back, is there, can you take a sample and manipulate it and get it into the system effectively at this point?
Oh, yeah. So people, are you talking about sample handling where they scoop it in? Yeah. So that's the typical way these missions work, is they have some sort of sampling handling. It's a robotic shovel, a scoop. They have drills now on Mars. So for Europa, they're sort of like a buzzsaw type thing that they're proposing to alternative oil and get these things. They're difficult because sometimes you get surprised by the behavior of the soils. The Phoenix lander was surprised by that. The soil was a lot stickier than they were anticipating. It wasn't dry. It had some permafrost in it, some other properties. So it actually would stick on the gratings that they were trying to get into. So the sample handling and extraction is actually quite a big thing, and it's always a worry. So people try and do standoff instruments, a lot of spectroscopic instruments. The current Mars rover has a ton of standoff instruments now that does remote libs and fluorescence. So they can try and measure things without actually requiring samples. But microfluidics applications certainly need samples.
Yeah.
Yeah.
I know often just like getting some particulate in your sample can just destroy an instrument pretty much in microfluidics if it's not robust against that, and
yeah,
you extract out what you want without losing all of it to the atmosphere in a sense. So
exactly. So that's one of the things we're working on, is using ultrasonics to separate the organics from these mineral grains.
yeah.
So you can filter out, remove the mineral grains, and get them into your instruments without any clogging. And that is a process as well.
Okay.
Yeah.
I just had one quick question, and then I'm going to jump over to the chat questions. But there's a lot of, we see a lot of companies jumping up, a lot of cool technologies coming about using droplets, and I think of the droplet technology as a place of getting a sample and encapsulating it and kind of storing it in an environment and such. But there's a lot of science behind it in terms of efficient droplet generation, surfactants to keep things stable, to do what you want with them. Has NASA or LMT looked at droplets as a potential microfluidics tool to use in space, and assessed kind of the how promising that is as a tool that could go up there? Or maybe somebody's already done it. I'm just not aware.
I'm not aware.
I'm not aware of it either. Yeah. I say we've looked at it, in whenever a new science comes up that we're trying to develop a mission for and develop the technology for, we ask, what is the best way to do this? And my mind certainly goes generally among other technologies to droplet microfluidics. We haven't had the science application yet that's a great match to that. Now, I think as we begin to talk more about things like sequencing and looking at complex microbial communities in that context and other things, it might very naturally be the thing to do, but we haven't had occasion to use it yet. You could say we've been biding our time and sort of commercially and in academic labs, it's a technology that's gotten better and better and better over the years, and now is not just in academia, successful commercially. So we're ready to pounce on it and use it in whatever way is best. But so far we haven't had the magic overlap that made it, in our opinion, the right to do the particular missions we're planning.
Cool.
I'm interested in droplet microfluidics, but we haven't explored that yet. It does seem to have a lot of applications, especially for point of care diagnostics and other things.
Yeah. Mm-hmm. It's got its own family of challenges to get the whole thing going. I was curious if anybody had worked through that yet for space missions, but it'll be interesting to see how it goes. So let me jump over. We had a question from Casey. Can you use the external vacuum of space as the way of pulling the gas out of the system? So does the bubble trap vent to space?
Yeah, so the answer to the first part of the question is absolutely yes. You can use the vacuum of space if you're careful about it. Our microfluidic systems are designed to operate in 1 atmosphere, and they fly in a hermetic enclosure that's typically pressurized around 1 atmosphere. And that makes the testing much easier in the lab, because you don't have to try to make all your microfluidics work under conditions where water might boil and other things happen. And some of the components work fine and are totally leak tight due to surface tension of water, but if you pull a vacuum outside of them, they're not leak tight anymore. So, but if you need the vacuum for something, there are 2 things we use it for. We actually don't use it for the bubble traps because it's a bigger pressure drop than ideal for the bubble trap, but you hit on something important there. If you do pull a vacuum on the hydrophobic membrane with a sample or with some aqueous solution on the other side of the membrane, and do so for a while, of course, water vapor, not only trapped gas, but water vapor will go through that hydrophobic membrane. That's why Gore-Tex is great for boots and jackets. If you do that for a while, you can concentrate your sample. So you can pull out water, and if there happen to be any organic volatiles, of course, you'd lose those too, and gases, but all the things that are water soluble, like ions and larger molecular weight things, they stay behind. So we've built little vacuum powered concentrators where, in a matter of some hours, we can get a 100-fold or better than 100-fold concentration of the sample. And when we talk about going to look for life, that can be really important, because we think a lot of the molecules we might want to look for could be present at quite low concentrations if they're there on Enceladus Europa. So we would use the space vacuum to do that. The other thing we use it for, which some of you probably have done yourselves in microfluidics, is so-called priming. If you start with a completely dry microfluidic system and you'd like to fill it up with liquid everywhere, maybe you just start with buffer initially before you put your sample of interest into it, and you want no bubbles in any corners or anything, a great way to do that is to evacuate your entire microfluidic system and then backfill it with water. And the water or the buffer will rush in and fill every channel, every corner, every valve. And so we actually have set up systems where we would use the space vacuum to pull all the air out of our dry microfluidic system. They typically have to fly dry because if you go to Enceladus or Europa, for example, Europa takes between 2 and 5 years to get to. Enceladus takes more than 10 years. So you fly the whole microfluidic system dry, just like you would maybe package a point of care diagnostic, and don't get it wet till you're ready to start using it. So space vacuum's a nice way to wet everything out in preparation to start doing the analysis.
Okay.
Just comment to membranes, which can, actually pull out only gases and still keep, water.
Mm-hmm.
So they may be, beneficial for priming even, and other stuff, right?
Absolutely. Absolutely.
Okay.
Yeah, we use them a lot in various parts of our fluidic system.
Okay.
Yes.
Okay.
See, there was a question from Ben Hawkins for Nathan. Is the electrochemical sensor ion specific that you were showing?
No, it's a separation technology, so it's not ion specific. You identify ions by their arrival times during the separation. So specific ions will arrive at specific times. And sometimes the art of the electrophoresis in that system is actually finding the right background electrolyte that will allow you to separate all the ions. Not all background electrolytes and at all concentrations will separate equally. So depending on what you're looking for, you have to tune your background electrolyte properly.
Okay. Yeah, just thinking about the separations, you mentioned the CEMS as one of the tools, or I guess the MS is one of the other technologies that'll take full advantage of the vacuum, so that you don't have to send along a mass spec with a vacuum pump. No need for that, right?
Yeah, absolutely.
Okay. Let's see, for Tony, are organ-on-a-chip microfluidics useful to understand human responses in microgravity? You talked about the bone density loss. What about other organ-on-a-chip things?
Yeah, I think they're going to be very, very useful, and they are one of the technologies that we think should be a lot in future plans. We have been limited in doing our small satellite experiments, our CubeSats as they're called, to experiments where we can put our biological sample in a state of stasis and sitting around on the launch pad side the integrated payload for a period of quite a number of weeks. We did actually have some success in Greg Kovac's group at Stanford a number of years ago, actually. I think it was Tammy Dukas and Laurent Giovangrandi, and Nicole was there at the time. They had some success in getting cardiomyocytes to get into a state of stasis for at least a couple of weeks, where essentially they could get them to revive after that and not have to tend them much for a couple of weeks. But in general, as you know, if you work with mammalian cells and mammalian chips, putting it away for 6 weeks and hoping that fine just doesn't work out. With experiments that go to the International Space Station, and as we look forward to the Lunar Gateway that's in planning, those are cases where what's called late access. In other words, you can bring your sample on board the rocket the day before, or even hours before launch. I think that's going to let us move towards doing more organ-on-a-chip work. And it's been proposed on ISS, on the International Space Station, but I don't know that anyone's done it there. I think it's a great idea. It certainly would let us look at reduced gravity effects at the cellular level, of course, and sometimes that doesn't matter, but sometimes there are surprising effects, particularly to cells that are gravitationally sensitive, like bone cells, and in some configurations like muscle cells. And of course, it's also good for looking at radiation effects at the cellular level. So yeah, that'll be important.
Cool. I got the next one here from Aditya for Nathan, and kind of another push around the separation space, CE, like the microchip isotachophoresis can possibly be leveraged as a sample preparation technique, as it can offer a pre-concentration factor of greater than 1200. So not really a question, but I guess any comment around that as a tool for use up there, and maybe any issues with deploying it?
I think it would be great. Yeah, I appreciate the comment.
Yeah, that's good.
Too. Absolutely. I've long thought we should figure out how to do that. So I think Nathan and I are on the same page on that one. Yep.
See, is Juan here tonight? He's normally at the meeting. So
Yeah.
See, a big ITP guy, right? Yeah.
Yep.
Cool.
Absolutely.
Yeah, and then I saw Andre Babineau has a question about planet landings out there. The question is there a planned landing on Europa, and if so, what's the timeline look like?
Yeah, so there is a plan of sorts. There was something called the Europa Landers Science Definition Team that convened a number of years ago and put together a quite detailed plan for a lander and what science it should do, and many other details. And in fact, there has been some NASA funding over several years. We've had some of, and Nathan has worked with us on some of those projects to develop the instruments that might go to Europa on a lander and be used to search for life. So the ones that we've worked at Ames are some of the ones that I showed you today, the one that combines capillary electrophoresis, the one that has an array of ion sensing electrodes, and also the microscope, which is being developed at Nathan's company. The plan does not go as far yet as congressional and full NASA approval. In other words, the budget's not allocated. This will be what's called a flagship mission. As you can imagine, putting a lander on the surface of Europa will be quite expensive. The Europa Clipper mission is, I believe, going forward if there aren't further funding hitches. That is an orbiter of Europa, and so it'll give us a lot of reconnaissance and tell us a lot about Europa that could pave the way for a future lander. There are still studies underway at JPL to do more planning for the Europa lander mission, and so it is actively in work in planning stages, but it's not approved yet, and it's not funded to go. I think my perspective is it's a matter of when there's a Europa lander, not if, but when could be. Probably it won't launch any sooner than 6 years from now, if I were to just take a guess. But it could be 15 years from now. And so a lot of that is, the politics, because it's very expensive, is wrapped up in that, and NASA's priorities. But I think we'll see it, and maybe closer to the 6-year time horizon than the 15 if things go well.
Yeah, and as Aditya is asking, and I assume that the plan will be to go after the subsurface ice water under the ocean there.
The first one actually is just going to go a ways underneath the surface ice, and as Nathan said, use something sort of like a buzzsaw, a grinder, an abrasion tool to scrape some ice that's far enough below the outer surface that it's not so badly radiation damaged. Because if you know something about Europa, you might know there's lots and lots of radiation on the surface because it's inside Jupiter's radiation belt. And that's a matter of the practicality of getting through a couple hundred meters of ice, which is a heck of a drill. Or people immediately think, is there something radioactive I can use and basically melt my way through? And of course, that's been thought about, but it's hard. It's hard to get through that much ice. The reason why the surface is not necessarily a lost cause is that Europa, Enceladus for sure, and Europa very probably, based on some images, has vents where there are fissures in the ice, and some of the ocean water is able to basically find its way all the way to the surface and spray up in a so-called cryovolcano. Basically, think of Old Faithful in really low temperatures at Yellowstone, where the water freezes to ice before it has a chance to go far. And so there are places well understood on Enceladus and places posited on Europa where you can find relatively fresh ice on the surface that was ocean water not too long ago. And so that would be the goal. And I think the Europa Clipper orbiter will tell us where such places might be and how feasible it is to make a lander land basically where the snow has been falling, if you will, where that snow comes from an ice fissure and the ocean below.
Cool. And then, no, Dave Clegg, yeah, old buddy. He's got a question for Nathan. You got an interesting correlation to sample collection and introduction to microfluidic systems in space. How much of what is done, the sampling and processing in a space environment, translated to applications on Earth, especially for things that we just don't want to be exposed to? So, like hazardous situations.
Yeah, I think there is a lot of applications for that. I'm not sure, I'm not aware of a lot of people currently applying that yet. That is one of the things we're interested in. We sort of have an environmental monitoring bent, but that could also be taken into situations where the environment is so hazardous you don't want a person. But it would be translate very well if you had a robot out there that could essentially serve as your Mars rover and throw things into the system. Then you don't have to worry so much. That could be a high radiation environment, chemically contaminated area, or other hazard.
Or Tom Dashiell's office.
Yeah, after the wrong piece of mail. Yep.
Yeah, so those would be great examples where NASA programs lead to kind of things that we just take for granted nowadays, right? It's kind of cool. Let's see, Tony, what's the life expectancy of CubeSat missions for these kind of things?
Yeah, interesting. So for certain deployments where we've had deployments at lower, relatively lower altitudes, the life expectancy, I think the quickest one we've done was up 60 days. And that's because it was deployed significantly below the International Space Station. You're doing them by a mission that's going to the ISS to resupply it, for example, which we've done once. You deploy it well below the ISS so that as the CubeSat orbit decays, it doesn't run into the ISS. That would be really bad. And because a lot of our biology missions have only taken a week or a couple of weeks to grow the cultures and see what's happening and what are the microgravity effects, that's perfectly fine. The longest one we've done so far is still up there, and it's got an expectancy of, it was 22 years estimated life expectancy before it would burn up. Now, that was the one that and I worked on, Oreos, and we were do the biology measurements for 6 months and the measurements of the organic biomarkers and so on for 8 months. And the satellite continued to function. Our partners at Santa Clara University, students and staff there run the contacts with the satellite. Initially during the beginning of the mission, multiple times a day. And then after the science is done, they may contact it weekly or monthly. They were able to still make contact and verify that a lot of the sensors, batteries, solar panels, et cetera, were still working for 5 years. The design time months, and you try to build in a factor of 2, but we were really happy that it lasted for 5 years. This also gave us some information about the leak rates because a pressure sensor showed us that the pressure, I forget the exact rate, but it loses something like 1% of an atmosphere, a couple of % of an atmosphere per year after hermetic container, which is, for ultra high vacuum technology, no big deal. But so that's kind of the range. BioSentinel actually will be in orbit for maybe 7.5 billion years until the sun engulfs and other stuff in our vicinity. But it's designed to operate for at best maybe 18 months.
See, one from Alexander for Nathan. He's wanted to see if you could be a little more specific on POC, environmental and water monitoring testing opportunities in space. So it's like looking for where these things could be used outside of.
Oh, sure. I was thinking in my talk more about a lot of the technologies we develop would translate to those applications on Earth very well. But there are water monitoring testing opportunities in space. The ISS, for instance, has a program to actually develop instruments to test for their water on board. Any closed system water cycle, like on board ships or submarines or in a space station, has to keep its drinking water supply clean and make sure it's clean. So one of the things they are really interested in the ISS is, apart from heavy metals and such, bacterial contamination or even fungal or protist contamination. So they have to monitor that very carefully so people don't get sick. One of the issues is that they actually take water samples now. So they go up there, they take water samples, they return them. So that you can imagine the lag time in the water quality measurements. That's one position there. Environmental monitoring applications, I would classify that into planetary exploration. If you were go to Europa or such, and you have this meltwater generated from the ice, you could monitor that. But point of care, it hasn't come up yet in NASA, but I think it's got to come up soon if they're really serious about sending astronauts into space and for very long durations and keeping them on the moon. They may not have to look for transmissible diseases so much if they keep quarantines before they launch up and running, but they do need to monitor a lot of health conditions that evolve from the space environment. There's a lot of oxidative stress that they'll suffer. They might want to look for cancer markers and other things like this, or actually monitor bone density by looking at calcium ions in the blood or urine. So I am going to assume that in the not too near future, or not too distant future, POC applications will start to be called for.
Yeah, let me just say that there was actually a lot of consideration of POC sensors a number of years ago, and for various reasons, prioritization and politics being among them, it sort of was ramped way down in 2006 or so, with the idea that it would be brought back before there's a long-term mission. So I think I agree with Nathan. It will be back again. There was for those of you who might know, the iStat point of care sensor for blood chemistry, and I think this version that has gone to the International Space Station was the one that's used for measuring chemical species in urine. That one actually did go to ISS and was tested for a period of quite a few months. So that's the one sort of commercial off-the-shelf POC sensor I know of that has been to space, to the ISS, but it has not been a highly active field, and I think it will become so again.
It was one of the really early ones commercialized, actually, right?
It was, yeah, 96 or 95, something like that. Yeah.
Simple, and I remember the challenges around like getting the cost down and such, but they've managed to make it through. It's a great story.
Yeah,
now, right?
He said it was the double-sided sticky tape was the real secret. We all know how
like always
PSA is, right, to plastic fluidics, and I talked to Laux once, who led that company in the early days, and he said it was all about the tape, man. That was it.
I think in some of them that's still true. Oh boy. Okay, so I'll try to combine a couple here. So Casey had a question about when you're putting together the CubeSat, what's really the biggest challenge, and what's the enemy end up being? Is it the mass or the volume when you're trying to package everything together?
Okay, so there's sort of
10 kilograms in a 5 kilogram box.
Exactly. So the enemy there is that we all of those of us who are sort of science minded want to put as many science capabilities as we can into that small box, and the wise engineers in the group want to put in less so that the chances of it all fitting in there are better, and eventually there's some compromise struck. But really, I would say, because these satellites are so small and because they have more stuff than you might reasonably think you could fit in a can that small, a lot of the challenge of integration is putting everything in really carefully and not breaking anything as it goes in. And as you might think, you have to really develop a process flow for exactly what order you're going to do this, and practice it a lot. And sometimes the way you do it first is completely wrong, and you learn because something breaks, and then you integrate it again, and you learn some more, and you integrate again. And so these things, by the time we fly them, we've probably done integrations 10 times, but in some of them 20 or 30 times. Others, if it gets too close to the wire, not enough times. But it is fitting everything in there and making sure you don't cause a leak or crack a trace on a circuit board or anything like that. So all of that translates into challenges when you're doing the design. We tend to design things thinking, what do they all look like, and can they all spatially fit in the can? We don't always think about how will we physically build them up part by part, subsystem by subsystem, and insert them into the can. Well, nowadays we do, but when we did the first ones, we didn't think properly in those terms. Now we actually will catch things and change our designs earlier in the process, because particularly the mechanical engineers will say, hey, I've integrated these things before. we have to change something here because this is going to be impossible to integrate, even though you can fit it all in this 3D model. So yeah, that's it.
So you got one for Nathan from Andrew. In what ways do you see the electrical impedance spectroscopy to offer improvements as a detection measurement?
Well, there's a number of things we're exploring. Being a small company, I can't talk about them too much. But one of the things, if you're looking at larger molecules and separation technologies like electrophoresis, if you're using the multi-band detection limits for C4D or just direct electrical impedance, you can learn a lot more about the analytes you're looking at.
Yeah.
There's also interesting applications telling the difference between water and ice phases if you're looking for water on airplane wings. There's a lot of, it opens up, whenever you can add more information to something, you can learn a lot more, I guess is the general thing. And we're doing electrical impedance measurements single point for our instruments right now. And if you can do it at 6 points, you can learn a lot more.
Yeah. I can mention one that I worked on at Stanford, not for space, but we were able to develop a system that could, first of all, could differentiate different types of cells, red cells, white cells, platelets as they flowed past the electrodes. Not a big surprise there, but then our real goal, and eventually we were able to do it, but only by using multiple frequency wavelets and some other good tricks, we could tell platelets that were in their normal so-called resting state, which is good, and platelets that are in called the activated state, where the platelets are basically ready to start making a clot or do something else that's, well, good if you're bleeding and you need to patch a damage in a blood vessel, but not good if they're building up on the walls of your arteries. And so multi-frequency impedance spectroscopy let us differentiate and count how many of a blood sample. Cool.
Okay, so I think I'm gonna try to capture Grebs Thompson's question in kind of a wrap up here, but to keep us on schedule for Thomas and team, one more kind of question in the sense that with I know that Nathan had mentioned opportunities for space technologies as a company. Do you see with the likes of Bezos and Branson and Musk all generating this commercial space flight options and going to different planets in a private sector? Do you see that that's going to boost some opportunities for people working in opportunities for getting on space missions as opposed to spending time working on the ground? And as I assume that's going to outside.
Uh-oh.
Experiments there. We'll have the microbreweries out there as one opportunity for companies. The question is, will there be what other opportunities do you see now that we're going to have the commercial space flights coming up?
I guess it depends on the level of the commercial space flights. So if we're talking about the space tourists that go up for a few minutes and come back down, I don't see a lot of opportunity for companies like ours to benefit. But if we're talking about actually colonizing Mars, then there's a lot of opportunities for us to benefit. That's where I think SpaceX would really be interested in a point of care device to monitor health. I think they would be right on top of that. I think they would be needing and really interested in process monitoring for closed cycle water. I really think that they would be interested in instrumenting a lot, when the microfluidics allows you to instrument a lot in a small package. So I think that on the horizon represents a potential opportunity. For the near term, where it's just space tourism, I don't see a lot. Now the CubeSat applications are different. Getting rides on with CubeSats is a different opportunity. And Tony can speak more to that. There's a lot of companies that are trying to make swarms of CubeSats to do a various number of things, and I'm sure that helps them a lot.
Yeah, certainly Earth observation is one where significant numbers of CubeSats have been deployed, and you can have your own specialized Earth observation network that covers a lot of places on the globe, or covers the whole globe with good frequency for a cost that's amazingly low compared to what it would have cost 30 years ago, if you can do it with CubeSats, if they provide the functionality you need. And that's really been doing that sort of mission is in the commercial domain already. It doesn't require sending people to space, of course.
I think the next phase we're going to move over into kind of the job board. Is that right, Thomas? I want to one question though, leading up to that. I know both you guys had mentioned that kind of had different backgrounds before coming into this, and at some point made a transition into doing research into space. And you both see it as a great career, career direction. Essentially, you have a lot of students, postdocs, early stage folks that attend. Want to see if just kind of get a feeling for what drove you that direction a little bit, but then also how do you figure out what's the best way if someone is interested in going that direction? How do you prepare for something like that, or where's the best way to make a contact to find opportunities there? Either one. I think as a company or a national group.
Yeah.
Right?
You want to
I was going to say, Nathan, you've done this more recently, and also you were much more deliberate about it, I would say, than I was, in that you went after an NPP. Why didn't you start?
Sure. I guess in graduate school, I became interested in astrobiology. Well, I've always been interested in it, but it was a very interesting time, and a lot of the research that we were doing just happened to align really well with astrobiology. So there was a sort of a natural segue. Taking that transition from graduate school into a postdoc, if you're interested in space applications for anything you're doing, I would highly recommend applying for a NASA postdoctoral fellowship. The best way to do that from a practical point of view is to find researchers at NASA who are doing things that you're interested in, or that you think you might want to contribute to, and cold calling them, getting in contact with them, and talking about if there's any sort of overlap. Before you turn in your application even, just talk to people. Just as with everything, applications that go into a system might get lost or ignored if somebody's not looking for them. So as a practical point of view, go out, shake hands, make that phone call, and talk to people. And then it's a great program because you're free labor to your advisor, essentially. They don't have to pay for anything. So they want somebody to work for them. But it also comes with a lot of freedom as well. It comes with a free travel stipend and a lot of other things like that. It has a lot of perks. So if you're interested in making that transition into space applications, or you want an academic route like that, getting your feet wet at NASA is a great way. It's also a great opportunity if you want to go into a commercial sector, because you make connections and you learn a lot. You meet a lot of great people at a NASA center. They know a lot. You get to network a lot, and that's very important to the practical point of view, is actually understanding the space, so that the space of space that you're working in, the people, what they want, what they need, and how things are done. And that will prepare you to be more successful in a space venture if you want to start one or join one. And from there, there's a lot of opportunities to work at NASA. But then if you wanted to start your own company or join a company, there are also growing opportunities. Companies like Blue Origin and SpaceX and Virgin Galactic have really sort of sparked an interest in the community that funds small companies. So there's increasing interest in funding small companies and getting them off the ground to do interesting space applications. As such, there's a lot of opportunities to either start or join a small company if that's your interest. But I think going into space, I would highly recommend one of these NASA postdoctoral fellowship programs. Also, if you're still a graduate student or an early career or postdoc, I think there's also the JPL Summer Planetary School. And if you apply to that, I think they tend to take people who are more already in that space. But if you have any applications there, that's also a great program to get into. It's not as long as a postdoctoral program. It's a summer essentially, but that's also a great opportunity.
Okay, I guess with that, Thomas, you got sounds like you've got a few companies who have openings, so a chance for it. I think just thanks again for Nathan and Tony for giving us great talk and getting lots of great conversation going there. And I'll hand it over for the job postings.
Yeah, thanks a lot, Don. So Samir is going to present a few jobs here that companies are recruiting for.
Yes. So thanks again to Nathan and Tony. And so job board, we have a job VP of Microfluidics from CFOX, and we don't have a representative from CFOX today, but Thomas will put their contact and the link to the job in the chat. And then next we have Thermo Fisher, and I believe Savat or Kavat. Yeah. Sorry. If
No
I
worries.
pronounced it wrong.
No worries. Do you want me to say a couple words?
Yes,
if we have time?
please.
Okay, well, thank you. Thank you, the BAM team. Thank you for the discussion today. I'm a senior manager over here at Thermo Fisher Scientific in the Genetic Sciences division. We have a pretty large R&D presence here in the Bay Area. We have a couple sites, and we are working on a lot of clinical diagnostic product development, qPCR, Sanger sequencers, microarray type of projects. So I have 2 roles open in my group, and we have another 2 roles open in one of the other groups in the larger organization. So most of them are related to fluidics, molecular biology, microfabrication. So I think you might find some of these roles interesting. So if you find them interesting, and just feel free to reach out to me. I'll be happy to talk to you and give you more details. And thank you for the opportunity.
Of course, thank you. And next we have Pin Cow from Nutcracker.
Hi, thanks. It's great to be talking to everyone. It's also great to see some familiar names. It's been a while. So Don and Tony. I don't know whether you remember me from many, many years ago.
Oh, yeah.
Absolutely.
Yep, definitely.
Good to see you guys.
Yeah.
So let's see. My name is Pin Cow. I'm VP of Systems Engineering at Nutcracker Therapeutics. Let me say a few words about our company. We are an early stage biotech company developing mRNA using our own internally developed microfluidic system. mRNA, I think a lot of people know this from the COVID vaccines. It's obviously being applied as a vaccine for COVID. But what we're doing is developing the technology to apply it to other diseases. We have a couple of exciting, what I think is our exciting opportunities. We have a principal systems design engineer with a lot of mechanical engineering focus. This would be a lead role to help out with bringing out our first platform. And we also have another role. This is a senior/staff mechanical design engineer. More on the design side. Again, a very hands-on person is what we're looking for. We actually have a third rec that we're going to open up again for a mechanical engineer. For all 3 roles, these would be for working on our generation 1 and as well as our generation 2 systems. And they're quite exciting with what we're working on and how it could impact healthcare. Specifically, all these roles are for working on the instrumentation to control our microfluidics and fluidic systems. So if you know anybody that might have experience in that area, or if you're interested, please contact me. My contact info is up on the screen. Okay.
Thank you. And I hope you're staying for the networking session so that people can maybe connect with you and talk to you. And so with that, thanks again. This was amazing. Thank you, Don, for moderating, and thanks to Tony and Nathan, and clap if you can, and then go to the breakout sessions. See ya.